Well, good morning, good afternoon, good evening to all of you that are joining from all across the world, making, in fact, IMMUNOTALK global. My name is Florent Ginoux, and it's my pleasure and honor today to host and introduce our global IMMUNOTALK speaker today, Professor José Carlos Alves-Filho. Before doing this, a reminder that we will have also Prof. Anne O'Gara next week on October 26th, so stay tuned and please do join us also on October 26th. So again, today we will have Professor José Carlos Alves-Filho from University of São Paulo. He's an associate professor leading the Inflammation and Pain Laboratory and a principal investigator in the Center for Research in Inflammatory Disease, the CRID, at Ribeirão Preto Medical School in the University of São Paulo. So Professor Alves-Filho is from Ribeirão Preto in Brazil. He's Brazilian. He graduated in pharmacy and biochemistry at the Pontifical Catholic University of Rio Grande do Sul in 2002. He received his PhD in pharmacology from the University of São Paulo in 2007. In 2008, he joined the University of Glasgow as a postdoctoral research associate in immunology at the Institute of Infection, Immunity, and Inflammation. In 2011, he returned to the University of São Paulo as a professor in the Department of Pharmacology at Ribeirão Preto Medical School, where he's now an associate professor since 2019. So as we will see later in this talk, his work focuses in the field of immunoregulation, studying innate immune dysfunction in sepsis, long-term immune dysfunction after sepsis, as well as molecular mechanism modulating plasticity and differentiation of effector and regulatory immune cells. I just want to mention that also that he's an elected affiliate member of the Brazilian Academy of Science in 2012. So again, really thanks so much for accepting our invitation and for giving us a global IMMUNOTALK, which will be untitled, Understanding Sepsis-Induced Immunosuppression. But before we start, as a tradition, we always have a question for you, and this will be about your career. And the question will be, could you share one of the most impactful decisions in your scientific career, please?
Thank you, Florent, for the introduction and for the opportunity to be here in this fantastic initiative that is the Global IMMUNOTALKs. And I'm really glad to be here sharing with the opportunity to share what we have been doing in our lab. And my most impactful decision in my career was in 2011, when I was a research associate in University of Glasgow, and I decided to move back to Brazil to start my own lab. So in that time, the situation in Brazil, the economic situation in Brazil was pretty good, and we could get a grant. One of the grants was to create the Center for Research in Inflammatory Disease. That was very important. And this was funded by an important agency in São Paulo State called FAPESP. So that was very important to me to have my own lab. And the situation nowadays in Brazil, it's in all aspects, but in terms of science, is very dramatic. The budget for research is reducing a lot, especially for research outside of São Paulo State. So I hope we will have an election for president in 2 weeks, end of October. And this is an opportunity for all of us Brazilians to have to change the situations and try to have a better turn of science since 2023. So that's my most important decision to move back. I don't regret so far, despite the situation, but it's become difficult now.
Really, thank you for sharing this with us, and we're looking forward to your talk. And you can please start to share your slide. And thank you so much for again accepting this invitation.
Do you see my screen?
Yes, I do.
So as Florent introduced, today I share with you one of the line of investigation in my lab, where we try to understand the sepsis, the mechanism involved in sepsis-induced immunosuppression. The research interest in my lab, as Florent just said, is to understand the molecular basis of immune cell activation, function, plasticity, particularly in these two groups of cells, the Tregs versus Th17 cells, and inflammatory macrophages versus regulatory macrophages. So these two groups of cells are very important and are under the mechanism of many inflammatory diseases. And we focus the role, we have been investigating the role of these cells and apply this in the context of autoimmune diseases and also in the context of sepsis-induced immunosuppression. That's the topic of my talk today. So sepsis, by definition, is a systemic inflammatory response that results from an uncontrolled infection. Infection that can be induced by any kind of pathogen, bacteria, fungal, protozoa, or virus. Well, these statistics before SARS-CoV-2 infection probably changed a little bit because we know that SARS-CoV-2 also is able to induce a cytokine storm, is a kind of sepsis as well. But in general, 90% of the cases of sepsis are related to bacterial infection. In terms of the immune response in sepsis, the immune response in sepsis is very complex. That's why it's so difficult to treat patients. Okay, so we can didactically divide the response, the immune response in sepsis by different steps. So the first step is the more characterized one that we call systemic inflammatory response, and this is characterized by systemic inflammation with high levels of systemic inflammatory cytokines, including TNF, IL-1 beta, IL-6, and also some alarmins like MHGB1. Okay, it's exactly at this phase that we have tissue damage, and also the tissue damage leads to organ dysfunction. And then eventually many patients die during this phase of sepsis. But if the patients start to treatment in the hospital in an early stage point. Before that, actually, during my PhD and my postdoc, we have contributed and in this to the knowledge of this phase, showing that the development of systemic inflammation is due, at least in part, to an impairment of neutrophil migration to the site of infection. And we showed that the systemic activation of neutrophils via toll-like receptors induce the downregulation of chemokine receptors in neutrophils and then avoid them to migrate into the site of infection. And this, of course, if you don't have migration of neutrophils into the site of infection, so we cannot control the primary site of infection, and then the infection spread, and then you have systemic inflammation. We also showed that IL-33, a cytokine that can modulate neutrophil and try to rescue the downregulation of chemokine receptors in neutrophils. But if patients survive to this acute phase, a very dangerous acute phase of sepsis, they start a second phase called compensatory anti-inflammatory response syndrome. That's characterized by the production of anti-inflammatory cytokines, mainly IL-10, and also, for example, antagonists of the receptor, IL-1 receptor, that try to compensate the inflammatory response, reducing the inflammation. And is in this phase also that start activation of many cells important for the tissue repair. And then many patients can survive from sepsis and then can discharge from the hospital. However, the problem doesn't stop here. For the last 20 years, many clinical studies have been showing that patients that survive from sepsis, they develop a late immune dysfunction. That's characterized by late death due to persistent immunosuppression and recurrent infections. Okay. For example, some examples, this study published in 2014 compared patients that discharged from hospital, septic surviving patients, and also patients that did not were there by sepsis, but other illness situations. And then in this study, they followed the patients for 1 year after hospital discharge. And we can see very clear here that patients that survive from sepsis, they have 6 times more frequency of new infection than patients that do not get sepsis. And also, these infections are much more severe, because this is another graph showing the cumulative frequency of rehospitalization by septic patients, surviving septic patients, due to infection. So they go to the hospital much more frequent than patients that did not get sepsis due to new infections. In another study, and this is interesting because they looked for 5 years after hospital discharge, okay? And they compared the general survival of septic surviving patients compared to the general population. And we can see very clear here that the survival curve of patients that survive from sepsis is much lower than the survival of the general population, especially during the first 6 months after hospital discharge. So my lab was interested in understand the mechanism underlying this high susceptibility to new infections and understand that this immunodysfunction after sepsis episode. To do that, so we needed a good experimental model that try to mimic the clinical scenario to have to later extrapolate and to do the translational research to the human scenario. So to do so, we employed the 2 different models and combined these 2 different models. So we induced sepsis by using the gold standard model of sepsis called the cecal ligation puncture. This model consists in open the peritoneal cavity and expose the sacrum and make some punctures in the sacrum, allowing the leak of the extracellular, the cecal content into the peritoneal cavity. So after that, we close the peritoneal cavity and wait them develop sepsis. And we treat them with what we call a basic therapy, that's antibiotic plus fluid replacement, wait them recover from sepsis, and then after they recover, we induce a secondary infection. When we use another place, we use the pneumonia model using opportunistic pathogens. So this is the characterization of the model. So when we induce CLP, they start to develop sepsis. And when we look at the mice 6 hours after sepsis induction, we can see that they have bacteremia. They also have systemic inflammation. We can see that they have high levels of TNF, high levels of I1 beta, high levels of IL-6. And also, when we look at the blood pressure, we found that these mice, they also have a reduction of blood pressure. So these all parameters characterize a severe sepsis. And then is exactly at this time point when they have already severe sepsis, mimicking what happened in patients that they go to the hospital with sepsis and then they start to be treated. And then we started the treatment with what we call basic therapy. So it's antibiotics and fluid replacement every 6 hours during 4 days. Okay? If you don't do any treatment in these mice, they will die 100% up to 2 to 3 days. But if you treat them with this basic therapy protocol, we could rescue some of them, around 40 to 50%, depending on the experiment. And this is exactly what happened in the clinical scenario. So patients that develop a septic shock, so around 40 to 50% survive from that. When we look at the general aspect of these mice that are surviving, so we look at for the biochemical parameters of organ damage and dysfunction, we can see that at 6 hours, when they start the treatment, when they have the acute infection and acute inflammation there, they have many parameters related to liver, to heart, to kidney. So we have increased parameters of tissue damage and dysfunction. But after treatment and they start to recover, so we can see a reduction of this parameter. And we know that, as I showed before, after day 3, they don't die anymore. So, and they completely recover this parameter of organ damage and dysfunction. We also looked for bacterial clearance, and we can still see some bacteria in the blood until day 7, but on day 15 and on day 30, after sepsis induction, they don't have a signal of infection anymore. And they also recover the body weight, so that they lost during the acute phase of sepsis. So if these mice are in the hospital, so they will be discharged there. So they have been cured from acute sepsis. And is this exactly with these mice that survive after 3 days, and we wait them to recover? That will work later on. So one experiment that we did, we challenged these sepsis-surviving mice 15 or 30 days, and we did recently 90 days after sepsis induction with opportunistic infection. We used, for instance, Legionella pneumophila, gram-negative bacteria, that normally is not able to kill naive control mice, but the same amount of bacteria when we injected intranasally in 15 or 30 days after sepsis induction, these mice die 100%. And this is not just related to bacteria infection. If you use fungal opportunistic infection, Aspergillus, we can also see high mortality in sepsis-surviving mice. Instead, in naive mice, we don't see any mortality. And also, this is it's not just a problem related to infection. We also found that the immunosurveillance to control tumor growth is also affected. So we used a tumor growth model using B16 murine melanoma cells injected here in the back skin, and then in the subcutaneously. And we can see very clear that mice that survive from sepsis, if you injected tumor cells 15 days after sepsis induction, we can see that the tumor cells grow faster in sepsis-surviving mice. So as soon as we have optimized this model, we published this model, and then later we started so we had a model that we believed that we can go now to study the mechanism related to sepsis-induced immunosuppression. And then, unfortunately, I don't have time to show all the story that we have been doing during the last 10 years of my lab. But I resume part of that in this scheme of this cartoon. So when we have sepsis, we know that sepsis induces systemic inflammation. And systemic inflammation induces tissue damage, organ damage, and eventually mice and patients die. But we know that if you treat mice or treat patients with antibiotics and also fluid replacement and other pharmacological approaches, we know that patients 40 to 50% can survive. And what we show is that patients that survive, we found increased production of IL-33 after the acute infection, okay? And we showed that also that we reported that IL-33 is important to induce the expansion and activation of innate type 2 lymphocytes. That's important to produce IL-13, and this cytokine is important to induce the differentiation of M2 macrophages. And we showed that this pathway is important because M2 or regulatory macrophages, they produce many growth factors and also tissue healing factors. That's important to induce wound healing and content balance the damage caused by the systemic inflammation. However, we showed that IL-33 together with IL-10 released by M2 macrophages, IL-33, we still don't know the source, but together they work together and they use the expansion and the differentiation expansion of regulatory T cells, and this leads to immunosuppression. Okay, and this we published a few years ago. We published this study was done by my postdoc Daniel Nascimento, and I don't go too deep into the details, but if you want to know a little bit more for the details, please go to this study. But another question that we could answer in this paper, apart from the source of IL-33, was that we know that macrophages, M2 macrophages, despite they are able to produce IL-10, they need a constant stimulation. It's not just a kind of differentiation, but they need a constant activation to produce IL-10. And we found that IL-10 was increased and kept it high for several days after sepsis induction. So, and then we also noticed that when we isolate macrophages from septic-surviving mice, and we did in vitro killing assay for with Legionella pneumophila assay. We found that macrophages from septic mice, they are not able to kill bacteria as naive macrophage do. So suggesting that apart from the Treg expansion, we also have innate dysfunction in macrophages. And this is related to, at least in part, to inability to kill bacteria. And then we started to search for a potential endogenous ligand that could affect the production of IL-10 in macrophages. And we know that adenosine is a very important molecule. Intracellular is important for the synthesis of DNA, RNA, and also for the metabolic energy. But in the extracellular compartment, adenosine can act and activate, can bind and activate 4 different kinds of receptors. And by doing that, specifically by activating the H2A and H2B receptor, they are able to inhibit effector T cell activity. They also are able to enhance Treg activity, induce Tregs, and also inhibit NK and KT cells, and also promote type 2 activation of IPCs, a type 2 activation of macrophages. All of them are features that many other groups also have been characterized in mice that survive from sepsis. And the source of adenosine, extracellular source of adenosine, is mainly from the hydrolysis of ATP, extracellular ATP, by by the activity of sequential hydrolysis of ATP by 2 different ectonucleotidases, CD39 and CD73. And when we look at the literature, we found it's a very old paper published in 2000, actually, showing that that there is a very high level of adenosine in patients, and this is a prognosis of outcome for septic patients with severe sepsis. And also another study by the group of Professor Simon Robinson and Haskell, they showed that the deficiency of CD39 deficiency enhance or is harmful for the survival, suggesting that activation of CD39 and production of adenosine by CD39 is important for reduce inflammation and attenuate inflammation in sepsis. Okay, so we measure it. So decided to measure the amount of adenosine in the blood and the plasma of septic surviving mice. And to do that, we used our model that we have standardized. So we induced sepsis by CLP, treated them with 3 days with antibiotics, and then we harvest the blood from different days until 75 days after sepsis. And then we analyzed we measured the adenosine by HPLC. We can see very clear here that the amount of adenosine on day 1 and here in the graph is reduced during the acute phase of sepsis. So probably this is one of the causes to high inflammation in the acute phase, because we actually are losing one important anti-inflammatory mediator. But for our surprise, we found that after day 3, we have an increased accumulation of adenosine in the blood of sepsis-surviving mice, with peak in between 45 days, between 15 to 45 days. And this is the time that we used before to induce secondary infection, and we knew that, and we found that mice that survive from sepsis at this time point, they are very susceptible to secondary new infections. As I mentioned, so the amount, the extracellular source of adenosine, and the main extracellular source of adenosine comes from the hydrolysis of ATP by CD39. And then adenosine binds to different receptors, especially NH2A. So activating a signaling, intracellular signaling pathway that's important for the production of IL-10. So, and IL-10 that leads to the anti-inflammatory, most effects of the anti-inflammatory effect from adenosine. So we asked to see if adenosine is playing a role, if it's increased after sepsis, so in sepsis-surviving mice, but we checked if there is a role of this adenosine, this increased amount of adenosine in sepsis-surviving mice. And to do that, so we used a protocol, a similar model. So we induced sepsis, vive sepsis, treated them with antibiotic for 3 days. And after that, we started on day 3 to treatment with the antagonist of the HOA receptor, the adenosine receptor HOA. We treated daily from day 3 until day 15. And after that, we stopped treatment, and then we did the secondary infection by challenge mice intranasally with Aspergillus or Legionella pneumophila. And as we can see here, as shown before, so vehicle-treated septic-surviving mice, they are highly susceptible to secondary infection induced by Aspergillus and also by Legionella pneumophila. But if you treat mice with HOA antagonist that block the receptor and avoid the signaling of adenosine, we could partially rescue the susceptibility of these mice, suggesting that adenosine via activation of HOA is important to induce, at least in part, the immunodysfunction followed in sepsis-surviving mice. We also did this using knockout mice, and we found similar results. And then we checked the ability to control the infection, because we know that the high susceptibility of sepsis-surviving mice to infection is because they cannot control the infection. We can see very clear here that the high amount have very amount of bacteria in the lung and also in the spleen that spread the infection in sepsis-surviving mice, in white-type sepsis-surviving mice. But if you treat them with the HOA antagonist that improve the survival, we also could see that we could recover the ability of these mice to control the infection. We then explored the role of CD39 as a source of adenosine. To do that, we used exactly the same protocol, but now we treated mice from day 3 until day 15 with CD39 inhibitor. And then after that, challenge them with Aspergillus and pneumonia, Legionella pneumophila. But before the challenge, we actually harvest blood from these mice. We killed mice and harvest blood for the quantification of adenosine. As we can see, as shown previously, when sepsis-surviving mice on day 15, they have accumulation of adenosine. Okay, and if you treat these mice with inhibitor of CD39, or if you use knockout mice, CD39 total knockout mice, we could avoid this accumulation. Okay, suggesting that the accumulation of extracellular adenosine comes from the extracellular hydrolysis of ATP. And then we look at for the importance of this in the establishment of sepsis-induced immunosuppression. And we can see very clear that, again, if you block or if we use knockout mice also for CD39, we can partially rescue the susceptibility of sepsis-surviving mice to secondary infection. We can see here for Aspergillus and also here for Legionella pneumothorax. So, adenosine is important, is accumulating in sepsis-surviving mice, and then via activation of HOA receptor induce the immunodysfunction. And this adenosine comes especially from the hydrolysis of extracellular ATP by the activity of CD39. So our next question was to understand which cells are the source of adenosine or express high levels of CD39. We know that CD39 is expressed in many different leukocytes, especially the regulatory T cells. Macrophages also can express. So we did a high-dimensional flow cytometry to characterize the expression of CD39 in different populations from spleen cells. And here we can see the expression, the degree of expression of CD39 expression in naive mice or in sepsis-surviving mice 15 days after sepsis induction. And for our surprise, when we look at here regulatory T cells, we can see that the regulatory T cells express CD39, but this cell population was not the population that expressed the highest level. But we found another population here that we can find this population in naive mice that is expanded in sepsis-surviving mice. And when we look at back to the type of cells, we found that there was a subpopulation of B cells. This for us was a surprise. And then we tried to characterize which subtype of B cells are expressing high levels of CD39. So we did again high-dimensional flow cytometry, and we found that a population here, so that express high levels of CD138, that's a marker for plasmablast population, is the population that express high levels of CD39. We can see this population in naive mice, and this population is expanded actually in sepsis-surviving mice. Okay, so we then look at the literature, and actually we found some studies in the literature that gave support to our find. This one not specifically related to CD39, but there is this study showing that from Rothstein group, showing that actually the expression of CD73 in this population of B cells is important to induce immunosuppression via production of adenosine. And also this study showing that there is a population of that they call it here regulatory B cells that also express high levels of CD39. But in these two studies, they did not characterize exactly which subpopulation of B cells that express CD39, and different for us that we showed that is plasmablast cells. So we next try to investigate the importance of B cells, and most important, the importance of CD39 on B cells for the sepsis-induced immunosuppression. And we used different tools to do that. And one of the tools that we used was these mice, the me knockout mice. These mice, they have a problem in the maturation of B cells, and then they don't have mature B cells in the circulation, in the periphery. And then we induced the model, we induced sepsis, treated them with antibiotics, and look at the amount of adenosine 15 days after sepsis induction. As we found before, so we have accumulation of adenosine, and now, for our surprise, mice that don't have mature B cells in the periphery, they don't have the accumulation of CD39. And when we challenged these mice with the Legionella pneumophila, the me knockout mice that survive from sepsis, we found again that these mice are more resistant to secondary infection, and this is related to the better control of infection locally and systemically. So, but in that time, we didn't have here in the lab CD39 floxed mice to see the real importance of expression of CD39 in B cells. So we tried and we used this approach, try to understand the specific role of CD39 expressed on B cells for the induction of sepsis-induced immunosuppression. And we took the advantage of the me knockout mice that we know that they don't have B cells in the periphery. And then we transfer bone marrow, white type bone marrow cells or knockout bone marrow cells to these mice, okay? And then we know that after that they have, so after they recover from the bone marrow transplantation, the B cells in the periphery of these mice, so we call it the CD39 knockout me chimera mice, the B cells from these mice don't express, or most of the cells don't express CD39. And this is true when we look at the expression of CD39 and the CD39 knockout me chimeras. We found a reduction that the B cells express very low levels compared to the knockout mice. And then when we look at the induced sepsis, and then we look at the amount of adenosine, again, we see accumulation 15 days, and now with this strategy, we don't see accumulation of adenosine anymore. So so all this data show that B cells actually play a key role in the production and the accumulation of adenosine in sepsis-surviving mice, and this is important for the induction of sepsis-induced immunosuppression. But how? How B cells can induce the immunosuppression? We know that they are important for the production of adenosine, but what's the importance of this process for the immunosuppression? To answer this question, we did a very simple experiment. We treated, actually, we harvested B cells from naive mice or from septic mice 15 days after sepsis induction. And we co-culture these cells with naive macrophages in vitro, okay, in the presence of Legionella pneumophila. And 2 days after, we quantified the amount of the bacteria as a bacterial killing assay. And also we measured the production of IL-10 as a readout for the activation, the signaling of adenosine. Normally, we found that if we do this only with macrophages without culture of B cells, this is the normal amount of bacteria that we found after 2 days of exposure to bacteria. And the culture of naive B cells, okay, even though if they are white type or knockout B cells for CD39, we don't see difference in the ability to kill bacteria. But if we do culture with the septic B cells from white type mice, now we can see that the septic B cells can induce a signaling that impair the ability of macrophages to kill bacteria. And this was avoided when the B cells don't express CD39. We played with the other hand of the process. So we used the white type B cells, naive or septic B cells, but now we used macrophages that don't express the HOA receptor. And if the macrophage, so again, if you do culture with the septic B cells, so we can see an impaired ability to kill bacteria. But if the macrophages don't express the receptor of HOA, we could rescue the ability of these cells to kill bacteria. And this was related to the production of IL-10. So if you look at IL-10 as a readout of IL-10 signaling, we can see very clear that when we do the culture of septic B cells with macrophages, we can see very high levels of IL-10. And this is avoided if the macrophages don't express HOA receptor, suggesting that IL-10, the source of IL-10 there comes from macrophages that are activated by adenosine. We are, of course, we played with the other protocols here to reach the conclusion, but the conclusion is that CD39 derived adenosine by septic B cells impair microbial killing in macrophages via activation of HOA receptor. Then we did an in vivo experiment. So we moved it back to our in vivo model, and now we use it a specific knockout mice that don't express HOA receptor only in myeloid cells. So we use the same protocol, we induce sepsis, treat them with antibiotics, and 15 days later, we challenged them with Legionella pneumophila, and we can see here that again, if the mice are knockout for HOA only in myeloid cells, they are more resistant to secondary infection. And this was related to the ability to control the infection. And then again, we can see very high levels of IL-10 in the lung of these mice. And then if we don't have signaling of HOA in myeloid cells, we don't see high levels of IL-10. So I don't have time to show all the data. We also explored how B cells are able to produce adenosine. It's of course via hydrolysis of ATP by CD39, but we showed that there is also a metabolic change in septic B cells, and they produce very high amount of ATP as well. So they produce the substrate, and they also express the enzyme to produce the product that is adenosine. But we also look at patients. So we try to translate our data to the clinical scenario, and we harvest blood from septic patients in the later time points in the hospital. And then we look at for the expression of CD39 in CD19 B cells. And we can see very clear that B cells, and then I don't have time to show all the data, but in patients, in septic patients, we also characterize these as plasma blast cells. So they express high levels of CD39. And when we measure the amount of adenosine in the blood, we can see very high levels, especially if you stratificate them in sepsis or in severe sepsis, and patients with severe sepsis, they have higher levels of adenosine. And finally, when we look at we did also killing ability. So the killing assay of human macrophages culture with B cells isolated from septic patients. So we can see here, so when you use the macrophages from normal healthy macrophages, we can see that they are able to kill bacteria. This is the amount of bacteria. But if you do culture with the septic B cells, we can see that we found more bacteria there, suggesting that the culture with B cells impair the ability of macrophages, human macrophages also to kill the bacteria. If you treat this culture with the HOA antagonist, if you add in the culture, we could completely improve the ability of macrophage to kill the bacteria. And if you do the other way around, so if you add in the culture a TPase, apirase actually, that's a as an enzyme, a TPase enzyme similar to CD39, we could even found less ability to kill the bacteria, suggesting that we have even more adenosine in the culture, and this completely avoided the ability of macrophages to kill the bacteria. And this again was correlated with the production of IL-10. So the co-culture of macrophages with septic B cells enhance or increase the ability of macrophages to produce IL-10, and this is even upregulated by the addition of apirase in the culture. As a conclusion of this study, we showed that sepsis-induced expansion of a subtype of B cells, a plasmablast B cells, that express high levels of CD39. I didn't have time to show here, but we found that there is a metabolic reprogramming of septic B cells. They are much more glycolytic, and they produce a lot of ATP. So they produce, as I said before, the product, and they also have high levels of the enzyme to hydrolyze ATP into adenosine. And adenosine activates HA receptor in macrophages, inducing the production of IL-10, that there is an autocrine and paracrine effect activating IL-10 receptor that reduce the ability of macrophages to kill bacteria, and this is important for the immunosuppression. Okay, and this study was published last year, again by my postdoc Daniele, and you can go there to have more details about this study. And then going back to the cartoon that I used before to explain the different phases of sepsis, of immune response in sepsis. Now we know from different studies from the literature, but also we had contributed to that, showing that regulatory T cells, M2 macrophages, and also plasmablast expressing high levels of CD39 is very important for the establishment of the immunosuppression in mice and patients that survive from sepsis. And this is important for the induction or for the triggering of recurrent infections. I need to send thank you to my group, to the lab, to the students. All the students work very hard in these different studies, especially Danielle Nascimento doing her PhD and her postdoc, and also many collaborators that have helped with us by sharing mice and by sharing protocols and helping in different situations of the projects. And also the financial support, especially FAPESP, because without this financial support, we could survive. This is just to finalize, this is the Ribeirão Preto, the place where I am, and then this is the campus of the São Paulo University in the city of Ribeirão Preto. Is a city with about a little bit more than 500,000 people, and this is our campus, and this is the main building of the medical school where my lab is located. So thank you very much for the audience, and I'm happy to answer the questions in Twitter.